Morning students. Today we are going to discuss the remaining part of your previous lesson, fundamental units of life. First of all, let us recap the previous lesson in short. Yesterday you have learned about the definition of the cell, discovery of the cell, shape and sizes of the cell, types of cells, and cell theory. Let us discuss about the remaining points, structure and functions of the different organelles, differences between plant cells and animal cells, and some curious terms and facts related to the cell. Let us discuss one by one. Cell structures. Regarding the cell structures, cell is composed of some basic structures. Those are called cell organelles. Very first organelle is that a membrane. That is a plasma membrane. Plasma membrane is the covering of the cell that separates the contents of the cell from its external environment. It is a living part of the cell. It is very thin, delicate, elastic, and selectively permeable membrane. Plasma membrane is composed of lipid and protein. Basic function of this membrane is, as it is selectively permeable membrane, it allows the flow of limited substances in and out of the cell. Rather, plant cell contains one another covering, that is cell wall. Cell wall is non-living, thick and freely permeable covering made up of cellulose. Cell wall is available in eukaryotic plant cells and in prokaryotic cells. The basic functions of cell wall is like the following points. First, it determines the shape of the cell. It determines the shape of the cell. It protects the plasma membrane. It prevents desiccation of dryness in cells. It helps in the transport of various substances in and out of the cells. And the third important point regarding protoplasm. It is a jelly-like, viscous, colorless, semi-fluid substance in various cell organelles present in colloidal form. Protoplasm consists two parts, cytoplasm and nucleoplasm. Cytoplasm is that protoplasm found in between cell membrane and nucleus, while the protoplasm, when found inside the nucleus, that is called nucleoplasm. Or you can say cytoplasm is that part of protoplasm which surrounds the nucleus, or nucleoplasm is that part of the protoplasm which is located inside the nucleus. And basic functions of protoplasm acts as a store of vital chemicals like amino acids, proteins, sugars, etc. It is the site of some metabolic reactions. And the next organelle, Golgi body. Golgi body are also called Golgi complex or Golgi apparatus. It is first discovered by Camillo Golgi. It consists of membrane-bound structures arranged parallel to each other. And the basic functions of Golgi complex is like this. Golgi bodies store, modify, package and dispatch the substances, and they help in the formation of lysosomes. Now we should know about the ribosomes. These are extremely small, dense and round bodies interspersed in the cytoplasm. Ribosomes are made up of ribonucleic acid, RNA, and proteins. In prokaryotic cell, ribosomes are of 70S type, and in eukaryotic cells, ribosomes are of 80S types. Ribosome is responsible for protein synthesis. And the next organelle, mitochondria. Mitochondria are small, rod-shaped organelles. It is a double membrane structure. Outer membrane is smooth, whereas inner membrane is projected into numerous finger-like structures called cristae. Mitochondria contain their own DNA and ribosomes. That's why this is called semi-autonomous organelle. And its common functions like this. They provide energy for the vital activities of living cells. They store energy releases during reactions in the form of ATP, that is known as energy currency of the cell. Therefore, it is also called powerhouse of the cell. And the next organelle about centrosome and centrioles. Centrosome is found only in eukaryotic animal cells. It is not bounded by any membrane but consists of centrioles. Centrioles are hollow, cylindrical structures arranged at right angle to each other and made up of microtubules. Centrioles help in cell division and also help in the formation of cilia and flagella. And the turn goes to plastids. Plastids are present in most of the plant cells and absent in animal cells. They are usually spherical or discoidal in shape. These are double membrane bounded organelles. Plastids are also have its own DNA and ribosome. That's why this is also called semi-autonomous organelle. Plastids are of three types. Number 1, chloroplast. Number 2, chromoplast. Number 3, leucoplast. Chloroplast, green colored plastids containing chlorophyll. Chloroplasts manufacture food by the process of photosynthesis. And chromoplast, colorful plastids, they provide color to flowers and fruits. And the last, leucoplasts, colorless plastid, they help in the storage of various substances like starch, proteins and fats. And now turn comes to lysosomes. Lysosomes are formed by Golgi bodies. They are small, spherical, sac-like structures which contain several digestive enzymes enclosed in a membrane. They are found in eukaryotic cells, mostly in animals. And its common functions that help in digestion of foreign substances and worn out cell organelles. They provide protein against bacteria and virus. They help to keep the cell clean. During the digestion, in cellular metabolism, for example, when the gets damaged, lysosomes may burst and the enzymes digest their own cell. Therefore, Lysosomes are also known as suicidal bags of the cell. Very interesting fact and generally asked in the various exam. Why lysosome is called a suicidal bag? Now turn comes to vacuoles. Vacuoles means nothing but important for the plant cells. Vacuoles are empty. Vacuoles are liquid, solid filled in a membrane. In plant cells, vacuoles are large and permanent. In animal cells, vacuoles are small in size and temporary. In mature plant cell, it occupies 90% space of cell volume. Due to its size, other organelles including nucleus shift towards plasma membrane. Now discussion about endoplasmic reticulum. It is a network of tube-like structures from nuclear membrane to plasma membrane. It is absent in prokaryotic cells and matured RBCs of mammals. There are two types of endoplasmic reticulum. First, rough endoplasmic reticulum, and second one is a smooth endoplasmic reticulum. RER. In rough endoplasmic reticulum, ribosomes are present on the surface, and a smooth endoplasmic reticulum present where ribosomes are absent. And the common functions: it gives internal support to the cell, it helps in transport of various substances from nuclear membrane to plasma membrane or vice versa. It helps in the formation of plasma membrane and Golgi bodies. Rough endoplasmic reticulum helps in synthesis and transportation of proteins. A smooth endoplasmic reticulum helps in synthesis of transport of lipids. And the master organ of the cell is nucleus. Nucleus is dense and a spherical organelle. Nucleus is bounded by two membranes, both forming nuclear envelope. Nuclear envelope contains many pores known as nuclear pores. The fluid which is inside the nucleus is called nucleoplasm. Nucleus contains chromosomes and chromosomes contain genes which are the carriers of genetic information. And the common functions of nucleus is: nucleus control all the metabolic activities of the cell. Nucleus is the storehouse of genes. Without nucleus, cell can neither survive nor shows specialized activities. And now we are supposed to discuss about some curious terms and facts. In fact, plasma membrane perform various special functions like diffusion and osmosis. Diffusion is the process of movement of substances from its high concentration to the low concentration. During the respiration, carbon dioxide is given out, a waste product, and accumulates in higher concentration inside the cell compared to outside. Due to the difference in concentration inside and outside of the cell, carbon dioxide starts moving out of the cell. And the next term is osmosis. It is the process of movement of water from its high concentration to its low concentration through semi-permeable membrane. If the medium surrounding the cell has higher water concentration than in the cell, cell will gain water by osmosis. Such dilute solution is called hypotonic solution. Though water molecules pass across plasma membrane in both sides but more water will enter inside the cell. The cell is therefore swell up and increase in volume. This process is called endosmosis. If the medium surrounding the cell is exactly the same water concentration as inside the cell, there will no net movement of water across membrane, resulting in no change in size of cell. Such solution is called isotonic solution. If the medium surrounding the cell has lower water concentration than inside the cell will lose water through osmosis. Such concentrated solution is called hypertonic solution. This cell will therefore shrink and reduce in volume. This process is known as exosmosis. This is the end of this chapter. But before the end of this video, you are supposed to discuss the following questions. First question goes to you that name the organelle which is known as suicidal bag. Very clear to answer that lysosome. And name the organelle which is known as powerhouse of the cell. No one except mitochondria. Name the kitchen of the cell. No one except plastid or chloroplast. And now you are advised to write the differences between plant cell and animal cell, which is being discussed in this lesson. Thank you and have a nice day. Good morning, students.